For the ones that don't know me, my name is Marco Prado. I'm a professor at Western and a scientist at the Robarts Research Institute. I've been involved with a number of open science projects through the Rodent Cognition Core at BrainScan. And so, it was timely that we could get Dylan to come here and, well, not to come here, but to show up in the Zoom.
As close as we can get.
Yeah, as close as we can get, and present about open science to our community. And I'm going to say a few words about Dylan, and then I'll let him present. Dylan's talk is titled "The Open Future of Neuroscience Research and Innovation," and Dylan is actually this Open Science Alliance officer for the Tannenbaum Open Science Institute at the Montreal Neurological Institute. So it's located there. So this is part of a major effort to really increase transparency in science, and with that increase reproducibility and replicability in science. But there's a number of other implications for open science, including return to society. And I'm hoping Dylan is going to touch on all those different points. So Dylan really interfaces with the national and global open science communities in this role that he has, and is really promoting the uptake of open science tools and practices, particularly for the Canadian neuroscience community. He's got a background in neuroscience, health ethics, and law, and so he has a breadth of expertise that are needed to understand the challenges to really, sometimes not just do open science at your lab, but become an open science institution. So he has a lot of experience, has a lot of contacts in the open science world, and in this talk, he's obviously going to discuss what he thinks is going to be the future for neuroscience. So thank you, Dylan, again for presenting this to us today, and I'll let you take this away.
Perfect. Thank you very much, Marco. So the first thing I'm going to do for everybody is share a link in the chat. So I'm also going to share my screen once I've shared that link. If you click that link, what'll happen is it'll open up the Open Science Framework page where I've shared these slides. If you go down here, you can open up where the slides are shared. I am going to show you that this actually works. Download them directly and then open them, hopefully. Come on, PowerPoint, you can do it. Perfect. And then start my presentation. So I've shared it like this for a couple of different reasons. I don't need to introduce myself. Thank you very much for doing that, Marco. The main reason is that I have a lot of material to cover. There is far more to cover in open science, and even at the intersection of open science and neuroscience, than I could possibly do in 35 or 40 minutes. So by sharing these slides, hopefully you can download them. You can use them to make notes and use them for reference for the question period at the end. I am going to try to move through this material relatively quickly. However, there are also all sorts of hyperlinks included on the slides in the talk. So if you'd like to follow up with me or just follow up with those hyperlinks for more information, please go ahead. Little plug for OSF. I'm a huge fan of the Open Science Framework, and I highly suggest you check it out. So this talk is split into roughly 4 parts. I'm going to first address what the heck open science even is, talk about open science and neuroscience research, open science and open innovation, and then hopefully open it up for a significant question and answer period. So first of all, what is open science? Well, usually a talk on open science starts with a slide like this, some big word cloud or some list of activities that people have identified as relating to open science. But open science is much more than just a checklist. Open science, most fundamentally described, is the position that the most effective way to conduct discovery is through a maximum of transparency in the research process. Or, as Michael Nielsen put it better than I ever could, open science is the idea that scientific knowledge of all kinds should be openly shared as early as practical in the discovery process. If you haven't read Reinventing Discovery by Michael Nielsen, I highly suggest you check it out. It is the book that got me into open science, and even being 10 years old is probably the best and most accessible resource on it. Why are we talking about open science now? Why has it suddenly become a hugely important thing? Well, it's the intersection between openness and networked science. Networked science is simply the idea that science is now conducted in the context of computers and the internet. You know this. It's impossible to do science or escape from the necessity of using computers and the internet to conduct your research. Computational systems have allowed us to generate immense amounts of data, more than any single lab can ever possibly generate, and to analyze it in brand new ways. Interestingly as well, they're embedded in every single component of the scientific research process, which means that you either automatically or can generate records of every single component of the process, which is important for the replicability and reproducibility concerns that I'm going to talk about a little bit later. What you can do with computers is interlink information based on the relationships and create a data web, sometimes called a semantic web. It's sometimes called an interlinked commons, information commons. When you combine computers with the internet, what we have is the ability to amplify collective intelligence in a way that we have never before even dreamed of having access to. We can design architectures of attention to bring experts to exactly the questions that they're most likely to answer, and architectures of participation that allow them to actually contribute to the discovery process, regardless of the fact that they may be around the world and many time zones away. To just drill down a little bit more into each of those, we have the capacity to create a data web, this system of information where all of the components are interlinked based on their relationships with one another, rather than disparate and linked only tangentially through things like papers and citations. Some examples of minor data webs, not the full data webs, you can check out any of the Allen Brain Institute's atlases, the observatory. You can also check out the International Brain Lab, which I highly suggest. It's a group of 22 different labs that are working together to define brain circuitry. Collective intelligence is the ability to capitalize on group intelligence that emerges from collaboration, collective effort, and competition. You may think that competition is a bit of a strange thing to bring up in open science because there's this conception that it's all about lovey-dovey, but that is not the case. If you want to check out some resources about expert collaboration, I suggest you check out the neurodegeneration method development community happening on Protocols.io from a bunch of groups that are funded by CZI. You can also go locally right here at Western and check out the touchscreen cognition community. I'm a big fan of the touchscreen cognition community. I've been watching it very closely. You can also expand it beyond experts. You can crowdsource discovery. If you haven't heard of Galaxy Zoo or Foldit, I highly suggest you check them out. These are platforms where the public can actively contribute to answering scientific questions. If you want examples that are more specifically in line with neuroscience, I suggest you check out Mosaic and iWire. They're fantastic efforts that allow the public to trace neurons or fill in neurons and be able to analyze and characterize data in a way that no individual scientist ever possibly could. Open competitions are also absolutely fascinating. The idea of an open competition is that you compete to design a piece of software, for example, that completes a particular function. It could be characterizing brain areas. It could be figuring out how many CDs you can stuff into a box. It doesn't even particularly matter. The thing that makes it unique is that as soon as somebody posts a solution, it is openly available so that other people who are competing in the same competition can adapt it and use it and iterate upon it to be able to create new solutions and even better solutions. This type of competition is absolutely fascinating. I highly suggest you check it out. If you want an example that's more specifically related to neuroscience, I suggest you check out the Kaggle competition, the Trends Kaggle competition. To put this in a little bit of historical context, because I always like to, we talk about open science now, but the open science movement as it currently exists is really the second revolution in how scientific information is collected and shared. The first one, we have to go all the way back into the Enlightenment in the 17th century with the start of the scholarly journal and an increase in international trade. What this did was increase the record of scientific information and the rate at which it was shared. The result of this laid the basis for our modern world, our modern culture, all of the technology that we see around us today. It is still the system that we primarily use, led directly to the Industrial Revolution. The open science revolution is going to be equally as impactful. To give you a little bit more modern history of the open science movement, and it really started with what's described as the open access movement, what's been called the open access movement. This happened because the traditional way of spreading scientific information was through journals on paper. When this was the primary way of doing things, large publishers had an immense amount of marginal value to be able to contribute to the scientific process. They could coordinate review, edit submissions, and disseminate publications to exactly where they needed to go. This has been drastically changed by computers and the internet. We can now distribute scientific information at extremely low cost anywhere in the world, and yet there's still a push on the part of many publishers, certainly not all, but on many publishers, to maintain restricted access. This led to a significant strain on library budgets. I remember being in a faculty meeting at the Faculty of Law as a student representative when I was at Dalhousie University. And the shocking statistic that came up there was that after 3 years, they would no longer be able to buy books. When a library can no longer buy books, you know there's a problem, and anything that can excite librarians this much, you know, is worth paying attention to. This led to the open access movement. There's been an astounding growth in the various different components of the open access movement, especially the growth in preprints and especially during the COVID pandemic, which I'll be addressing a little bit later. It's led to prominent institutions like MIT and the University of California system ending their relationships or their contracts with large publishers like Elsevier. If you want more information on this, click that link and it'll take you to Spark. They are world leaders in this area. But we can extend the logic far past open access to publications. We can extend it to every single component of the research cycle. What this means is that we can share information about literature reviews, we can share information about data acquisition systems, we can share the data that we collect, the software that we use for analysis, really every single component within that cycle. And by doing so, we can create that interrelated data web that I described to you earlier. We have the opportunity to create a rich, open information ecosystem of all of these different components that can then, if it is truly open, be mined, remixed, adapted, and reused. The possibilities here are truly staggering. Moving on to neuroscience, I've given you a little bit of background. Why does neuroscience need open science, or why is it particularly relevant for neuroscience? I'm going to present 3 reasons. The first is that the social project of trying to understand the brain is absolutely enormous. We've got billions of neurons, each with potentially thousands of dynamic synaptic connections. These, at the molecular level, depends on thousands of genes and proteins which interact in ways that are largely either not well understood or not even known about. In scale, it makes the Human Genome Project look like child's play. The best we can do so far is create things like partial mesoscale connectomes in mice. If you want to learn more about this, check out the mouse connectome project. The one thing that I'm going to add in here is that the mouse connectome project never would have been possible if it wasn't for the openly shared data sets created by the Allen Institute. Without them, the amount of work required to create a mouse connectome would have been far too much for any lab to take on. What this, I hope, communicates to you is that with open data sharing and open sharing generally, we can raise the bar, we can set the pace, and we can iterate upon one another's work in a way that advances neuroscience in a way never before possible. If we don't take this type of approach, developing a similar map of the human brain at the micro level would be either a nearly impossible task or it would be something that would take decades. That might not be a problem if it wasn't for reason 2. Neurological disorders are one of, if not the major unmet medical need. I don't have to take you through all of these statistics, but they cost billions of dollars a year. There are millions of people in the US alone that are afflicted with them. The failure rate for treatments for Alzheimer's is above 99%. We're still using drugs or slight modifications on drugs to treat Parkinson's that were developed back in the 1950s. The state of affairs is so dire that a lot of major players have actually stepped out of the neuroscience game entirely. If you click that link, it says Pfizer, it'll take you to the page discussing the fact that Pfizer completely closed operations on its neuroscience branch. What's worse is that, as far as we can tell, there's not really any clear path forward. Because in order to have a clear path forward, we need to understand what these disorders are actually doing. We've made some great progress in that way, but in terms of having a whole picture that allows us to reliably generate treatments or diagnostic tools, we need better biomarkers, we need better patient and disease characterization. We need better targets and better tools. What we ultimately need is better neuroscience. And what I hope you take away from this talk is that the best way to craft a better neuroscience is to adopt open science and open sharing principles within neuroscience as a discipline, to bind together in the same way that the genetic and genomic communities did to advance the Human Genome Project. Without that type of community consensus, the human genome never would have been solved. Reason 3 is the replicability, reproducibility, and power crises that affect neuroscience. You can click on these links. Here are some excellent examples, but there's a feeling within the scientific community that there is indeed a reproducibility, replicability, and power crisis. The biggest problem here, of course, is that without relevant statistical power, without a large enough cohort to be able to source or to be able to sort out signal from noise, and without replicability of experiments, whether people can repeat the same experiments on different cohorts, or even reproducibility, where people can reproduce the same results from the data that you've collected, science as a method just doesn't work. This amazing thing created back in the Enlightenment that we call the scientific method just doesn't work. What it means is that we need to recreate the way that we're doing it. We're at an incredibly exciting time. You are at a point in history that is roughly equivalent to what was happening with Galileo and Newton when they were designing the modern scientific research system. It is up to you to be able to create it. Part of the problem here, of course, is that some people don't design studies very well, or p-hack or something along those lines, but that doesn't solve insufficient sample sizes. For something like that, you need to be able to combine data collected across many different experiments, which is much more easily done when people openly share the data that they create. And another major problem, as my friend Nicholas Stikov likes to say, is that the PDF is simply not enough. To be able to read a PDF and then accurately replicate every step in the research cycle, or to reproduce results, often is a non-starter. What we need to do is move beyond the idea of contributing to the literature through papers. Papers are still going to be massively central, important, and relevant, but we need to move beyond that and think about contributing to the information ecosystem, this data web or interlinked information commons, to be able to have sufficient power, enable replicability and reproducibility. A brief aside here, to step away from neuroscience for a second. The Organization for Economic Cooperation and Development and many other global leaders have said that open science has been absolutely critical for combating COVID-19. These are just some examples of open science practices that have been key to combating COVID-19. I could have also included, for example, the open hardware community that's been developing and distributing designs for face masks, testing kits, automated equipment for automated assays. It's a truly exciting time for open science. While COVID is a tragedy, what it has demonstrated is that it's only through open sharing and massive collaboration that we're going to be able to take on global crises. And if it's key for COVID, then why is it not key for the ongoing crisis of neurodegenerative disorders in terms of lives, suffering, and money? So I'll take you down to the neuro. What component of this are we working on? Because open science and neuroscience is not something that any one institute can possibly do alone. So what we've had to do is pick our targets to be able to advance where we can. The Neuro, as you may or may not know, was founded in 1934 by Wilder Penfield of the homunculus, and as a mixed clinical research institute. We started considering becoming an open science institute, so adopting open science at the institute level, back in 2014, driven by the possibilities that it held for discovery and the utter lack of new treatments for neurological disorders. We announced, after an 18-month consultation process to make sure that everybody was on board and everybody understood what we were doing, that we'd become an open science institute in 2016. To do this, we minted our open science principles, which you can find on the Neuro website, and made announcements in Science and Nature. After doing so, and excited by the possibilities that open science shared, or that open science held, we received a $20 million donation from the Tenenbaum Foundation for the creation of the Tenenbaum Open Science Institute, which is what brings me here today. There are a couple of different components of the Neuro's efforts. One, the first one I'm going to talk about, are the platforms that we've been developing. These platforms hit directly at the Neuro's dual mission of understanding the brain and bringing better treatments to our patients, because the Neuro is both a hospital as well as a research institute. So, identifying our comparative advantage, we designed the CBIG repository, which is the Clinical Biological Imaging and Genetic Repository. Click that link if you'd like more information. What it essentially is a shareable representation of patients, family members, and controls. When patients come in for a routine clinic visit, they're asked whether they want to participate. If they agree to participate, they can have samples, imaging, clinical information, and genetic information taken, put into the CBIG repository, which is then shared under a tiered access structure to make sure that we're sharing as much as possible, but not sharing anything that would be a risk to the privacy of the participants and patients. CBIG leads directly into the early drug discovery unit, where it takes the samples collected for CBIG and turns them into patient-specific stem cell lines, which you can use to create normal lines or neural organoids. This EDDU then shares the protocols that they use to develop these stem cell lines and shares the lines themselves. Right now, you have to know someone at the Neuro to get access to them. However, in November 2020, we're going to be launching the online portal, so keep your eyes there. The last thing I'll say is that the fact that these are patient-specific lines, we're not doing this in cancer cells or something along those lines, has excited industry. We have 8 ongoing industry collaborations, and we're able to raise $20 million in 2 years from a number of different sources. To take you through in a little bit more detail how CBIG and the EDDU are linked together, it starts out with patients because they're the center of our open science mission. During their normal clinic visit, through a consent process that's been specifically designed for CBIG, they're asked whether they want to contribute to the repository. If they agree, they have samples, imaging information, genetic information, and clinical information taken. Samples are sent to the EDDU. The EDDU then designs protocols in collaboration with industry to turn them into the best possible quality iPSC lines. It then performs its own research on it and shares a bunch of information there about it through the EDDU website, which can be accessed by people who want to use it for educational purposes, business purposes, coding purposes, medical purposes, or scientific research purposes. Importantly, the lines are also put back in CBIG, which can then share them upon request by someone from industry or an academic research institute. They will then be sent the iPSC lines, and as a component of being sent, they have to recontribute results back to CBIG so that it starts becoming an ever richer resource for everybody to use. As the final component, the results and papers and everything like that that's published are going to be made publicly available through the CBIG's website, so that the patients who originally contributed and are the core component of CBIG can see exactly what CBIG is doing. Open science, as you can see, is baked into each element of these platforms. It leads to increased collaboration. We've got more collaborations all the time. True patient involvement. It will accelerate discovery. It excites industry because this is a truly unique resource. And eventually, it's not happened yet, we're hoping to conduct new clinical trials at the Neuro, which is one of the things that the Neuro does best, and eventually leading to novel treatments to make our patients' lives ever better. Another major component, moving past the platform, is empowering community. This is something that is often not talked about enough in the open science world, but without an active community that's willing to engage in online spaces, engage in open science best practices, and work together, open science just doesn't work as well as it possibly could. You need to create a shared culture. Obviously, by the way, this picture was taken pre-COVID. You need to create a shared culture, a shared set of resources, and a shared vision. Because once you do that, you can coordinate efforts, integrate existing sharing systems. So instead of having to design the system all yourself, if people are already sharing on multiple different platforms, encourage them to adopt best practices. If they already are, then make sure that you integrate these systems with one another, make sure that what they're sharing is interoperable with one another's data, or protocols, or hardware designs, or anything that's involved in the research process. We need to create policies and incentives, and we need to educate both from the top down as well as enabling researchers to educate one another. If someone has taken a small step, then they need to be enabled to help others take that same small step. Many of the researchers at the Neuro, for example, are already sharing software toolboxes. They're sharing their papers through preprints and postprints to make them openly accessible, or publishing in open access journals. And they're also sharing data. It takes a significant amount of work to be able to make sure that these are all linked together to ensure interoperability, to set community norms and peer education, and to make sure that they're incentivized to do so in the ways that we can in sustainable ways. One of the things that we started doing recently in order to make sure that the grassroots efforts and the grassroots concerns at the Neuro are taken into account is to have pilot projects where a researcher will come and suggest, I think this is a better way to do open. And then we'll be able to give them small grants to be able to do a first pilot project and create documentation to allow others to do the same. The first ones that we're starting with is an open interactive neuroscience textbook, because part of our mission is also education. We have to educate the next generation if we want this to be successful. And a set of open source data standardization pipelines, because without standardized data, sharing it is of extremely limited use. So we believe that open science is the only way that we can truly advance and make the best neuroscience possible in the future. However, we recognize that it takes a world to understand the brain. We believe that open science, even if we're the only ones that do it, is going to be the best way for us to contribute to neuroscience and to the development of cures. But once other people, other institutes, other labs start adopting open science principles themselves, you have a multiplicative effect. You can actually achieve the kind of statistical power, for example, that's needed to be able to make truly groundbreaking results. This is why half of TOSI's mission, half of that 10 million that we received from the Tenenbaum Foundation, is intended to incentivize other institutes to adopt open science. We conduct seed projects to collaboratively build open science resources or to take a neuro open science solution and transplant it to other institutes. We do buy-in projects to help institutes figure out what exactly their open science approach is, and we hope to learn a lot from that. And if at the end they come up with a set of open science principles and an open science approach that is compatible with our own, we can offer some ongoing support. We're not alone, of course, in this. I would be remiss to give a presentation on open science and neuroscience without mentioning the Allen Institute for Brain Sciences. They're an unbelievable world leader in this area and have enabled an insane amount of downstream work. If you look at the papers linked to their atlases, they've got thousands and thousands of citations over the course of maybe a decade, and they're only going to get more all the time. We also have distributed networks working together and people building tools to enable distributed networks to work together. We've got national and international funding organizations that acknowledge the fact that it's going to take massive collaboration and ultimately openness to be able to make this mapping and modeling of the neural connectome possible. And then we've got individual institutes and labs who are also engaging in this. This is taking place all over the world, a little bit piecemeal at the moment, but hopefully more cohesive as we emerge into the future. What binds these efforts together, they don't have all exactly the same definition of open science, but what binds them together is a fundamental understanding that transparency and collaboration are going to be absolutely vital to developing a proper understanding of the brain and ultimately leading to treatments and cures for patients with neurological diseases and disorders. I would also be remiss if I didn't point out the fact that despite the rosy possibilities that I've been presenting so far, there are significant barriers to adopting open science. It requires culture change from the level of the individual researcher all the way up to global collaborations and everything in between. I don't need to go through each one of these barriers. I'm putting them there so that you can come back to them. You can ask me questions about them at the end, or you can do your own research into them. I'm more than happy to give you some resources about them. But what ties all of these together is that the system that existed before networked science and before the possibilities of open science and networked science don't necessarily mesh very well with what open science needs to truly thrive. And we need to solve each one of these components. We're doing what we can at the Neuro, but if other people have solutions in the spirit of openness, we would love to hear what you have to say. So why else should you care? Other than this rosy picture that I've been giving you of the creation of a beautiful information commons from which everyone can benefit. Well, open science is gaining worldwide attention, and that's especially been the case with COVID. Funders, from private funders like the Wellcome Trust, all the way through to the tri-agencies, are adopting open science relevant policies, whether this is an open access policy or a data management and sharing policy. They're happening more and more all the time. Oh, just check the chat. Oh, that's Marco. Thank you very much for handling that, Marco. Also, major funders are signing DORA. If you've never heard of DORA, the Declaration on Research Assessment, what it essentially is a declaration that we need to move past things like impact factor when judging the quality of science. We need to take a holistic view to the contribution that someone's made to the scientific endeavor, as opposed to being extremely narrowly focused on papers and the impact factor of the journals where they were published. Back in June, the tri-agencies signed DORA, so please keep an eye out for what's going to be coming out of that. They haven't instituted anything yet, but it will be coming. There's also more funding for open science tools. This is a link to CZI's funding for essential open source tools in open source software and science. I suggest you apply for developing open source tools as soon as the next funding call comes. But this is just to indicate that funders are recognizing that building tools is as important as publishing papers. Open science prizes are rapidly coming out. This is a link to the Neuro's Open Science Prize, but there's all sorts of different open science prizes that are coming out. There's also fantastic new recognition systems. So trying to work with the existing recognition system in science that primarily has to do with receiving grants and publishing papers. There are innovations like data papers and direct data publishing that can be put into your CV. There are journals specifically about publishing papers describing open source software that's being used in science. I should also let you know that if you do something like share data with your paper, there's good evidence to suggest that you're going to get higher citation counts. And if you publish a preprint before you do a paper, I can explain for those who don't know, if you want to ask me in the chat what a preprint is, you also get higher citation counts. The last thing I'll say in this category is that with network science, and especially with open science, the pace of innovation is only going to increase. I know you've all felt this. What rapid, timely sharing does for you is allows you to establish priority. You can, for example, publish a preprint and get a DOI that acts as a timestamp. That means that you're not going to get scooped, as it takes 2 years for your paper to be reviewed and then published by a given journal. Slide, please. There we go. So what I hope to have convinced you of in this talk is that open science, if we really want to advance open science as much as possible, must be the future if we truly want to gain a fundamental understanding of the brain. But I have to admit that open science does not have a single definition. This is because, this is the way that I think about it, there's a thin conception of open science and many, many thick conceptions of open science. The thin conception is generally what I've been talking to you about today, which is making sure that absolutely everything necessary to understand, replicate, and reproduce published findings is freely accessible, publicly accessible, and freely usable to the extent possible. Obviously, you can't just dump a bunch of personally identifiable information online, but to the extent possible, this should all be shared. A thick conception of open science, which is what you have to create at your lab level, at your institution level, at the country level, and at the global level, depends on a whole bunch of different considerations. Some people bring in considerations around diversity and inclusion, participatory design of experiments with participants, protecting patient privacy, citizen science, and much, much more. What I hope to stress to you is that it is up to you as the community of scientists at all of these different levels to define what your approach is and then advocate for it openly so that we can all have a good discussion about it. I'd like to finish off with a few words on open innovation. This is something that I get asked about a heck of a lot. So open innovation, like open science, doesn't have a settled definition. You can ask 10 different people and you'll get 11 different responses for what open innovation actually means. For us at the Neuro, based upon our principles, to be compatible with open science, innovation or translation, depending on which word you want to use, taking basic research and turning it into products and services that you can actually deliver to patients or the public or the research community itself, can't interfere with the ability of others to access and use the knowledge and tools that we create. In this context, and this is where I get a lot of questions, the traditional intellectual property practices at universities, such as patenting, can become an issue, especially if they restrict access to and use of the information and tools that are developed. Restricting access often happens when you have to keep information secret before getting a patent application, before submitting a patent application, because if you submit it before submitting an application, your invention, your idea is no longer patentable. And in terms of use, if the patent barrier prevents other people from being able to iteratively develop on top of it. I want to give you a few reasons not to be scared of this, though it may run counter to what you have been told as you advanced through your career. A lot of the time, traditional technology transfer doesn't work. There are a few highly publicized successes. However, there are many, many, many more, let's just call them not successes. This can often lead, patenting practices at universities can often lead to secrecy, can often lead to higher costs instead of savings. It can lead to research delays, creating a thicket of rights. We can talk about the CRISPR patent battle at the end if you're particularly interested. It's something that I'm fascinated with. And I mean, for all of these different reasons, if you'd like a more in-depth review and analysis of it, I shared a paper that I've been working on a preprint server for feedback from the global community. Please click and have a read. Reason two, we've gone with the negative reason, is the open innovation revolution. And for this, we need a little bit more history. So I love this story. This is the story of Linux, which is the open basis for nearly everything. If you have an Android phone, if you have a Chromebook, if you use Facebook, if you use Instagram or almost any other social media platform, or you want to look and see what people are using at major research institutions like CERN to run the Large Hadron Collider, it's all run on Linux. Linux, the Linux kernel, is a basic operating system. It was started in 1991 by Linus Torvalds when he was a master's student at the University of Helsinki. He was 21 years old and started building an operating system in the open with a group of like-minded individuals. 4 years later, Red Hat, which is the major company distributing Linux, was listed on the New York Stock Exchange for $3 billion and has a current valuation of around $30 billion. The Linux kernel has received contributions from almost 1,600 developers just in the time frame indicated there from when they began tracking. Open innovation generally, and Linux in particular, relies on designing innovation in a way that it can be modular so that you have tiny chunks that people can contribute to. Iterative innovation so that you can quickly modify tiny chunks and incorporate them into the product, and community development. In this context, openness is a feature, not a bug. Openness is absolutely required to make sure that the people who can contribute know about it and have the ability to make the modifications that they possibly can. This model is not restricted to software. It is now being applied in the hardware context in a bunch of different ways. I suggest you check out Opentrons. They're my favorite open hardware company and have been doing immense amounts for the COVID testing space since the large companies have developed and distribute tests have not been able to fill the gap. It's also being applied to healthcare innovation. I highly suggest you check out M4K Pharma and open source malaria. I'm almost done, I promise. So I want you to imagine this. There are three different scenarios here. You create a piece of neuroscientific research equipment. You don't patent it, you release it to the world, as well as the software upon which it runs. You collaborate with the worldwide scientific community or anybody who has the capacity to contribute. And then you sell finished product to the global community of users entirely in the absence of intellectual property. This is a beautiful vision, but is not science fiction. You can check out the UCLA Miniscope and how it's sold on OpenEFIS. The Miniscope is another one of my absolutely favorite open hardware examples. It also works for software. You can build it with the community, define standards with the community, and share it completely in the absence of restrictive intellectual property by assigning something like an open source license. I suggest you check out the Open Science Framework itself, which is the platform I use to share this, which is open source. You can also check out GitLab. Finally, and this is the area where we have the biggest complication when it comes to avoiding intellectual property, is the discovery of therapeutic compounds and medical devices. Medical devices is a problem as yet to be solved. However, in the therapeutic molecule context, at least for certain they're newly discovered or not yet FDA and Canada Health approved molecules, there are different models that don't rely on patenting to be able to bring it forward. I suggest you check out the M4K model, spearheaded by those who developed the Structural Genomics Consortium, Aled Edwards and the current head, or sorry, one of the current higher ups, Max Morgan. And they've also reached out into the neurodegenerative disease space. I highly suggest you check out M4ND, which is Medicine for Neurodegeneration. There was just a grant received by a McGill pharmacology lab to operate and to generate molecules to target the Parcan protein based largely upon this model. So to bring this to an end, an open approach to neuroscience, both at the research and innovation stage, is what we need to realize the full potential of networked science, of a world where the internet and computers are embedded to everything that we do. By adopting it, it's going to reveal things that are unexpected, beautiful, and life-changing. It's going to change how we do research and innovation forever. If you see the possibilities and agree with this vision, I ask you to do something simple. You don't have to adopt all of open science right away. What I ask you to do is to take one small step. It might be publishing in an open access journal. It might be sharing a protocol or beginning an open lab notebook through a blog. It might be taking a little bit of time to document a piece of code. Once you've done that, help others do the same. People are more likely to listen to their peers than someone that the higher ups bring in. And that's also the best way to have culture change. And then once you do it, advocate for what your vision of what open science can be, or advocate for what your vision of what open science can be. I can guarantee it's going to be coming to Western, to Canada, to the world. When it does, I highly encourage you to get involved, make your voice heard, go to the various different webinars or town halls that exist, and participate in its creation, because only through that can you have a stake and a say in the future of what open science is. That's the end. I'd like to thank you. So all the text, just one more second, Marco, all the text in this presentation I've released under a Creative Commons attribution license, which means you can take it, you can translate it, you can adapt it, you can take snippets, don't really care. Please feel more than free to do so. For information about the copyright in images and trademarks, however, or really any other questions you might have, you can feel free to contact me at dylan.roskams-edris@mcgill.ca. One of the reasons I did this is to demonstrate to you how much of a pain it can be when there are different intellectual property components associated with the elements that you can use to build something beautiful and new. So if you have any questions about that, please feel free. And with that, I'll end my presentation and stop sharing.